Hello, I'm Evan, and I have been chosen to do glycolysis. What is glycolysis? Glycolysis is the first step into the breakdown of glucose to extract energy for cellular metabolism. It's an anaerobic process, which means it doesn't require oxygen. It takes place in the cytoplasm of both prokaryotic and eukaryotic cells and begins with a 6-carbon ring-shaped structure of a single glucose molecule and ends with 2 molecules of a 3-carbon sugar called pyruvate. Glycolysis has 10 steps. The first step is a phosphorylation of glucose. This involves the transfer of phosphate from ADP to glucose, forming glucose 6-phosphate in the presence of a hexokinase enzyme. The second step is the isomerization of glucose 6-phosphate. In this step, glucose 6-phosphate is reversibly isomerized into fructose 6-phosphate. The third step of glycolysis is the phosphorylation of fructose 6-phosphate. In this step, fructose 6-phosphate is converted into fructose-1,6-biphosphate in the presence of the enzyme phosphofructokinase. The fourth step is the cleavage of fructose-1,6-diphosphate. The enzyme fructose diphosphate adductase catalyzes the cleavage of fructose-1,6-biphosphate between C3 and C4, resulting in 2 different triose phosphates. The fifth step is the isomerization of dihydroxyacetone phosphate. In this step, dihydroxyacetone is isomerized into glyceraldehyde 3-phosphate in the presence of an enzyme triose phosphate isomerase. The sixth step in glycolysis is the oxidative phosphorylation of glyceraldehyde 3-phosphate. The glyceraldehyde 3-phosphate is converted into 1,3-biphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Another thing that happens in the sixth step is NAD+ is reduced to coenzyme NADH by the hydrogen electron from glyceraldehyde 3-phosphate. The seventh step is the transfer of phosphate from 1,3-diphosphoglycerate to ADP. It involves the transfer of phosphate group from the 1,3-bisphosoglycerate to ADP by the enzyme phosphoglycerate kinase, thus producing ATP and 3-phosphoglycerate. The eighth step is the isomerization of 3-phosphoglycerate. It is due to the shift of the phosphoryl group from C3 to C2. The ninth step is dehydration of 2-phosphoglycerate. In this step, the 2-phosphoglycerate is dehydrated by the action of enolase to phosphopyruvate. The last step is the transfer of phosphate from phospholynyl pyruvate. Phospholynyl pyruvate is converted into an enol form of pyruvate by the enzyme pyruvate kinase. Overall, in the glycolysis process, you get a net total of 2 ADP and 2 NADH.